Hello, still Sir Remark for the second stage of photosynthesis, which is called as the carbon reactions or the Calvin cycle. Now, in carbon reactions or Calvin cycle, it makes use of the chemical energy produced from the light reactions to synthesize sugars or carbohydrates. In the carbon reactions, these happen in the stroma or the fluid that fills the space of the chloroplast, which contains its own DNA that is separate from the nucleus, enzymes, and other metabolic products. So this is the stroma. So the space that fills this one is called as the fluid that fills this one is called as the stroma. Now, the carbon reactions or the Calvin-Benson cycle or just the Calvin cycle or the C3 cycle requires carbon dioxide, ATP, and NADPH from the light reactions, ribulose 1,5-bisphosphate or commonly called as the RUBP, and the enzymes necessary for the different reactions. Overall, the cycle has three phases, which are the carbon fixation, reduction, regeneration of carbon dioxide acceptor, which is this one, the ribulose 1,5-bisphosphate. This is how the simplified figure of the cycle looks like. This cycle is already equivalent to 3 cycles, with 1 carbon dioxide entering every cycle. In the carbon fixation phase, carbon dioxide reacts with a 5-carbon compound called as ribulose 1,5-bisphosphate or RUBP via this enzyme called as the ribulose or ribulose bisphosphate carboxylase or ribulose bisphosphate oxygenase. Or the shorter name for this one is Rubisco. Now in the process, when the carbon dioxide reacts with RUBP, it forms an unstable product with 6 carbons which immediately cleaves into 2 molecules of 3-phosphoglycerate or 3-BPGA. Again, to show how efficient the cycle, in the figure, this is already equivalent to 3 cycles. So there is an incorporation of 3 molecules of CO2 to 3 molecules of RUBP, which makes 3 molecules of a 6-carbon compound, which immediately split into 6 molecules of 3-phosphoglycerate. In the reduction phase, the 6 molecules of 3-phosphoglycerate are going to be phosphorylated by the ATPs coming from the light reactions. There are 6 molecules of 3-phosphoglycerate, so we need 6 ATP molecules. These 6 ATPs give one of their phosphate groups to the 3-phosphoglycerate via this enzyme called as the 3-phosphoglycerate kinase, making 6 molecules of 1,3-bisphosphoglycerate, meaning they now have 2 phosphate groups attached to the to their molecules, specifically in the first and the third carbons. Still in the reduction phase, the 6 molecules of 1,3-bisphosphoglycerate are going to be reduced via the enzyme NADP glyceraldehyde 3-phosphatase, which oxidizes NADPH molecules coming from the light reactions, which gives the electrons to the 1,3-BPGA or 1,3-bisphosphoglycerate. In the end, there is a production of 6 molecules of G3P or glyceraldehyde 3-phosphate. The 6 molecules of glyceraldehyde 3-phosphates are going to be used to regenerate the initial 5-carbon compound, which is called as the RUBP. And the other G3P molecules are used to synthesize sugars in the form of sucrose or starch. So in the process, out of 6 G3P molecules, one G3P molecule will exit the cycle to be synthesized as in the form of sucrose or starch. And then the 5 remaining G3P molecules will be used to synthesize the 3 molecules of RUBP. In the RUBP regeneration phase, the G3P molecules undergo isomerization, condensation, hydrolysis, and phosphorylation to form RUBP molecules. Since we have 5 molecules of G3P, which contains 3 carbons each, overall we have 15 carbons. These 15 carbons are just going to reassemble, okay, to create 3 molecules which contain 5 carbons each. These 3 RUBP molecules are ready again to be fixed with 3 molecules of carbon dioxide that enter the cycle. To check your understanding of the Calvin cycle, answer the following questions. To produce 1 sucrose, how many turns of the cycle is or are needed? Also, in producing 1 sucrose, how many ATP molecules are used? And how many NADPH are used? Pause the video and think of the answer.